This video is going to be on an introduction to the blood gas. And to start out with, I have the acid-base equation written here. And as you can see, an acid is a substance that gives or donates a proton, and a base is a substance that receives or takes a proton. Now, during metabolic processes, just normal day-to-day events, the body produces a lot of hydrogen ions, a lot of protons. So the body on a constant day-to-day basis has to get rid of those protons. So it does this by either buffering those protons within the blood or excreting the protons from the body. Now, these protons can be measured or assessed by ClinPath tests, and it is measured as the blood pH. So now I'm going to write the bicarbonate buffer equation as an example of our blood gas reaction, or our acid-base reaction. So in this equation, carbon dioxide is an acid, and its conjugate base is bicarbonate. So in addition to the excretion of hydrogen ions produced from other acids, carbon dioxide is an acid, and it also has to be excreted from the body to maintain this livable blood pH. Carbon dioxide is a volatile acid, meaning that it can be a gas, and so it can be excreted by the lungs. But the non-volatile acids, like keto acids or lactic acids, those are not volatile, so they can't be exhaled. Those acids are primarily excreted by the kidneys. So next I'm going to explain the terminology that we use to describe changes in pH. So let's just say that the normal blood pH is about 7.35 to 7.45, and this range will depend on the species. So if there are excessive amounts of hydrogen ions, then that will make the blood pH decrease. And the process that causes these production of hydrogen ions, this is called an acidosis. So the acidosis is the process that leads to an actual change in blood pH. So when that blood pH gets outside of the reference interval or below the reference interval, then we call that an acidemia. Conversely, if there's a decrease in hydrogen ions, then that's called an alkalosis. So the alkalosis is the process that leads to an actual change in the blood pH. So if the blood pH is increased above the reference interval, then we call that an alkalemia. So if you see osis, then that's the process that actually leads to the emia, which is a deviation of the result from the reference interval. So now I'll go over the three main regulators of the blood pH. So these are the three main processes that help maintain blood pH within a livable range. And there's two broad categories. First, there's the metabolic component. So the kidneys and the blood buffers, those are considered metabolic regulators of the blood pH. And then the lungs are considered the respiratory regulators of blood pH. Now going to the kidneys, under day-to-day events, they normally act to excrete hydrogen ions and maintain bicarbonate ions. Going over to the blood buffers, there's lots of blood buffers, but the main blood buffer that we're going to discuss is the bicarbonate buffer system. And whatever buffer we're talking about, we're really talking about titrating hydrogen or some conjugate base which binds hydrogen ions to consume or titrate hydrogen. And then respiratory, that is when carbon dioxide is exhaled from the lungs. Now, there's also differences in how long these regulators take to actually manipulate the blood pH. The blood buffers takes the shortest amount of time. They only take seconds. The lungs takes a little bit longer, usually a couple of minutes to regulate the blood pH, and then the kidneys, they take the longest. They may take 3 to 4 days to actually regulate the blood pH. So now I'm going to talk more specifically about the bicarbonate buffer system. And I'm going to emphasize this system because it's the bicarbonate buffer system that's at the heart of blood gas interpretation. And that's because the three main components of blood gas analysis are within this bicarbonate buffer equilibrium equation. So, bicarbonate is the base in this equation, and carbon dioxide is the acid. And the hydrogen ions are measured on a blood gas instrument as the pH. And the pH is again under metabolic and respiratory control. The base, the bicarbonate, is either measured as bicarbonate on a blood gas analyzer or as TCO2, which is an estimate of bicarbonate that we'll see on a chemistry report. And this base, this bicarbonate, is under metabolic control. The acid, the carbon dioxide, we measure as PCO2, or the partial pressure of carbon dioxide, and for that you need a blood gas analyzer. And this tells us about our respiratory control of the buffer equation. And you'll certainly learn more about this equation when I talk about causes of respiratory acidosis and alkalosis. This is a summary slide of what tests we actually use to assess the metabolic and respiratory components of an acid-base status. And I have the three different tests that you can, or the three different panels that you can request from the ClinPath lab. So first, if you're ordering a blood gas, what you're going to use to assess the metabolic component is the pH and the bicarbonate. To assess the respiratory component, you're going to assess pH and the PCO2, or the partial pressure of carbon dioxide. If you request a serum or plasma chemistry, then you would look at the TCO2, the anion gap, and the chloride versus sodium to assess the metabolic component. And there isn't really a respiratory assessment that you can do with a serum or plasma chemistry. On the urinalysis, you can look at the urine pH to assess the metabolic component, and again, there's really nothing you can look at the urinalysis to assess the respiratory component. So in the subsequent blood gas and acid-base lectures and videos, you'll go through each one of these tests to help you identify what process and what disease is going on.